Hello, I hope you're doing well. Today we'll look at the Lac Operon. First, let's clarify some basics. What is a gene? You might have heard this a lot, right? It's in the genes. I got this gene from my mother. It's genetically passed on. A gene is nothing but a part of DNA which encodes for proteins. So let's further break that down. For that, we need to know the central dogma of molecular biology, which states that from DNA we get RNA, specifically your mRNA, and from RNA we get proteins. So, if a certain information is encoded in the DNA or the gene, that is not enough to express. The ultimate form or the ultimate unit of gene expression is protein. So a gene becomes expressed only if it becomes a protein. And DNA becomes RNA by the process of transcription. RNA to protein is called translation. And if DNA itself is replicating, it is replication. This is the central dogma of molecular biology. It basically states that this is the way in which genetic information flows. Now, what is an operon? An operon is a coordinated unit of genetic expression seen in lower organisms. By that I mean, eukaryotes or humans don't have operons, but we still study it in order to get a better understanding of regulation of gene expression. Having said that, let's try to understand the Lac operon. Simply put, it's a unit of it's a unit that works together in order to break down lactose. So lactose is the main player here, okay, and this operon is seen in bacteria, that is E. coli. Lactose is converted into allolactose. Slight difference in the structures, nothing much. So let's try, this is the operon itself. So let's try to understand the parts. It starts with a regulatory gene. Again, what is gene? It's just part of DNA. So you have your whole DNA, which I have represented like this. Okay, so if someone draws a rectangle like this, it is nothing but your DNA. So the operon starts with regulatory gene, which we denote with I. Now some people think I stands for inducer, but they are wrong. I actually stands for inhibition. Okay, this can come as an MCQ or something. So that's why I is a regulatory gene and it is a constitutional gene. For this, we need to understand the difference between a constitutional and an inducible gene. So inducible genes can be either inducible or constitutional. Constitutional means it is always expressed. There's nobody stopping it. And inducible means it is only expressed in certain conditions. We'll see what those conditions are. Okay. So now I'm saying the I gene is a constitutional gene, which means there's nobody coming to say, hey, I, now you make mRNA, now you make protein. No, I will keep doing its job. Okay. So that is about I. Next is the promoter. If you remember, promoter is a sequence which is usually upstream to the constitutional genes, and they help in the transcription of constitutional genes. Wait, if that's moving too fast, let's break it down. So here you have the main genes. Before the main genes, you have something called promoter. So if this is DNA, what is the next step? DNA to RNA is transcription, right? And transcription ke liye you need RNA polymerase. So what this promoter does is, if let's say this is RNA polymerase, the enzyme, the promoter directs the RNA polymerase to operator gene. It basically points out and says, hey, come here. You need to start transcription from here. Okay, so it promotes the transcription of structural genes. So regulatory gene, promoter, next is the operator gene. Operator gene is where the real work begins. The RNA polymerase actually binds to this site and begins its work. Then coming to the main structural genes, we have 3 of them represented by Z, Y and A. Z stands for beta galactosidase, Y stands for permease, and A is represented by thiogalactoside transacetylase, or simply galactoside acetylase. Okay, it's given in different books differently. I followed the standard textbooks. So these names can be a mouthful. Please say it out a couple of times so that you remember. Beta galactosidase, permease, and thiogalactoside transacetylase. Okay, this is the operon. Now we said that I is a regulatory gene. How is it a regulatory gene? It is also constitutional, right? So it will keep, this is the DNA means it will make one mRNA. mRNA is a straight line, right, usually. So let's say that the I gene made one mRNA. This mRNA should code for some protein because DNA to RNA, RNA to protein. So what protein comes out of this? It's actually something with 4 subunits, and this guy is called repressor. The word repressor might make you think of something which suppresses or inhibits, right? That's exactly what this is gonna do. This repressor will go and bind to the operator, and if the repressor is bound to operator, RNA polymerase cannot bind to operator. Okay, so just think of it like this. You have a repressor and then you have RNA polymerase. Both are trying to bind to the operator site, but repressor will stop RNA polymerase from binding. Okay, it's one angry little guy. Cool. So what did we understand here? This operon is gonna keep producing this repressor. So by default, none of these genes get transcribed. Okay, because it doesn't allow any of the genes to get transcribed. It doesn't allow RNA polymerase to bind to operator. By default, the operon is switched off. Okay. Now, who will come and switch this on? That's where our hero lactose or allolactose comes into the picture. Allolactose will come and bind to who? It will bind to the repressor. So it will be like, look, repressor, please leave the genes alone. I'm right here. Okay. So allolactose and repressor will form a complex which will detach from the operator gene. Okay. So let's say the repressor was here, and then let's say allolactose also came here and then said, hey, go away. So they do go away. And finally, RNA polymerase is able to come, bind to the operator gene and transcribe. Okay. In other words, we can call lactose, or strictly speaking allolactose, an inducer. What is an inducer? It promotes something. It promotes transcription at the genetic level. Not like your enzyme induction, and it's not like that. It's causing transcription of these genes. Okay. So what did we understand? Let's say there's no lactose. The regulatory gene is going to produce repressor. Repressor is going to bind to the operator gene and no transcription. But if there is lactose, that will bind to the regulator and the repressor lactose complex will go away. RNA polymerase can bind to the operator, transcription happens. Okay, so when you write it in the exam, you should write it under 2 headings. You should write switched off and switched on. Okay, write it separately, don't mix both together. Summarize whatever we've learned so far. First, allolactose inactivates the repressor. Correct, no. Let's say this repressor has gone and bind to the operator's site. Lactose will in turn bind to the repressor and take it away so that RNA polymerase can bind to the operating site, right? So allolactose inactivates the repressor. Next, bacteria switches on the operon only when lactose is present in the medium, which means if there is lactose, it will go bind to the repressor, which is always produced by the way, and it will allow RNA polymerase to bind and it will allow for transcription of the genes. If lactose is not there, none of this can happen. So the bacteria switches on the operon only when lactose is present in the medium. Third concept, lactose becomes allolactose, which is an inducer. Inducer means something which promotes a process at the genetic level. That's what is happening, no? It's binding to the repressor, it's allowing for RNA polymerase to bind. So it's inducing the operon. So it's an inducer. At the same time, it is also a co-repressor. So lactose and repressor kind of bind together, right? They have something going on with each other. So in that sense, lactose becomes a co-repressor. So don't get confused if they ask what is the co-repressor in Lac Operon? It is lactose. What is the inducer? That is also lactose, but more specifically allolactose. Okay? Answer an important question. We know that if there's lactose in the medium, the operon will switch on and bacteria will try to use it. If there's no lactose in the medium, the operon will be switched off. But what if there is both glucose and lactose in the medium? What happens then? For this, we need a bit more detailed understanding. ATP becomes cyclic AMP in the presence of adenylyl cyclase. This is something we know. Now, we have a player called CAP, and I'm drawing it in the shape of a cap. CAP stands for catabolite gene activator protein. Okay. Now this cyclic AMP or CAMP and this catabolite gene activator protein or CAP are best friends. Okay. CAP and CAMP form a complex like this together. And what this does is they will go bind to the operator and facilitate binding of RNA polymerase to this. In other words, let me just say that CAP-CAMP complex loves to switch on the operon. Okay, so like how lactose is an inducer, CAP-CAMP, this is CAP, this is CAMP, this also plays a role in switching on the operon. Okay, we'll keep it simple. Now where does glucose come in? Glucose will actually inhibit this. Let me draw it in red. Glucose inhibits adenylyl cyclase. What happens? As you can imagine, cyclic AMP decreases, CAP-CAMP complex decreases, so it is unable to bind to the operon, so transcription cannot happen effectively. In other words, glucose, if present, hates the operon, so it will switch it off. Okay, so that is something that's commonly asked. If both lactose and glucose is there in the medium, what happens? I hope this made sense. I would like to thank Dr. Alia for teaching this topic ever so beautifully to us that I'm able to say it 2 years from now. All the best for your exams. Bye.